Now I will call on our friend David Reiss. He's not only a friend of mine, but he's a friend of the Institute. He's spoken for us twice before, and we learned from him not only about science, a great deal about science and about the world we live in that we didn't know before. So David, if you'll come forward.
It's a little humbling to be preceded by two such elegant introductions. But let me express my appreciation to both Rabbis Luxemburg and Driesen for not only hosting us in this spot and at this time, but for together weaving an extraordinary intellectual tent in which we can have a discourse on a very big topic tonight. It seemed to me as I listened, both of you captured what Steve and I struggle with on an almost daily basis in the projects we're going to talk to you about, which is acknowledging, in this case, a very painful disarray of part facts, part fiction, but in the hopes, very much as both rabbis expressed, and in the conviction that we might find some order among this painful disarray and produce, Steve and I promised not to use any religious language. We were going to use just science. So in the course of our work that we can discover an order that will benefit a broad range of people. I also want to make one other thank you before I get started here, and that is to my partner, Steve Sumi. We are one of the longest running two-shoe acts or four-shoe acts in the business. We have lectured together in not only God-blessed places, but God-forsaken places. And it's also a chance for me to express to Steve directly how much I've learned from him over, I think, at least the 4 decades we've been collaborating on one piece of business or another. We are involved in a project that will seem familiar to you, but I think in the course of the time we present to you, you're going to see it from, I hope you'll see it from a very different angle. We're going to talk about severe adversity during fetal life and early childhood. And I think everybody in this room knows the tremendous risks that adversity during those two crucial developmental periods holds for the psychological development of children and adults. And it's also no secret that serious child maltreatment is highly prevalent. I'll show you some statistics about it, although statistics are a little hard to come by. It's one of the most prevalent risk factors for human ill health known. But we don't need research to say, let's do everything we can to reduce that to a minimum, and as soon as we can, after serious maltreatment occurs, to do everything we can to ameliorate it, for the child for sure, but also for the family. Plenty of research is being done in that area. That's not what we're going to talk to you about. What we're going to talk to you about is a thread of evidence, and you can start the story anywhere. I'm going to start it in the Boer War in the 1890s. A thread of evidence that suggests that these same adverse circumstances have significant and important impact on medical health, on heart disease, on stroke, on diabetes, on obesity and all its complications, possibly even some cancers, although we're not going to get into cancer tonight. And that although risks for these disorders that stem from early adverse circumstances may be manifest early with special testing, special investigation, often many of the adults who have experienced these dreadful circumstances arrive at adulthood without any obvious illness. And even in a world of the kind that the Aleinu prayer imagines, and we could develop effective intervention strategies, both to abate child maltreatment and to address its most serious consequences. There will be hundreds of millions of adults worldwide who will reach adulthood at risk for serious illness, for whom even the most comprehensive treatment programs never would have reached. Never would have reached. They live in the wrong country. They didn't have access to the service. The maltreatment was concealed. It is those adults who, having suffered these experiences as children and who reach adulthood subject to the serious risks of ill health, that Steve and I have been devoting the last 3 years to grappling with. And Steve will present to you some of the most extraordinary evidence in our portfolio now about how to understand that phenomenon. But what the two of us have been doing have been assembling leading scientists, primarily from Britain and from the US, and I'll perhaps explain to you why we're starting with that binational effort, to pool together a broad range of existing data and to develop a number of new ideas about how to approach this problem. We are at the very beginning. Some of the most basic questions in this field we're just getting to the point where we can ask them and we don't have answers. So what our intent is to do this evening is to tell you not so much about a line of science, although you hear about it, but to get you engaged in a social process among scientists and the assembly of scientists, and how such an assembly might work collaboratively to figure this out. So I said, you can start this story anywhere. So these are a line of British troops fighting in what's now called South Africa in the Second Boer War that spanned several years in the last decade of the 19th century. You might think of the Second World War as really World War III, the First World War as World War II, and this is World War I. A broad array of troops from the British Commonwealth fought the Boers, the Dutch-originated farming communities in South Africa, stimulated by a thirst for gold in South Africa that had been discovered in the 1860s, in an unbelievably brutal war that included concentration camps developed and set up by the British, in which untold thousands numbers of whites and blacks in South Africa perished under abysmal circumstances. So one could think about this as an adverse circumstance beyond imagination. But there was one sliver of good that came from this Boer War. Because Britain, in trying to mobilize hundreds of thousands of troops to fight in this war, discovered that a huge number of young men who came to their various recruiting offices were too skinny and too underweight and too undeveloped to fight. There are many preoccupations Britain might have had after this war, but this became one that led to something really quite splendid. Now I'm showing you the British county of Hertfordshire, just north of London, that little yellow dot. And in Hertfordshire, as in all counties, there was a chief midwife. But Ethel Margaret Burnside was quite different than most. Knowing about the scrawny state of British babies and being 6'1" herself and riding a bike all over the county, absolutely insisted that her midwives faithfully record the birth weight at the time of birth and at every single visit after. And she made sure that happened. And here's an example of one of the records. We'll shift south from Herefordshire to Southampton. This is giving you a clue as to why the United Kingdom is a crucial partner in this effort, where those birth records paid off. Southampton was the site of a medical school, and David Barker, whom I'm shown here, was a professor of medicine and epidemiology, and an extraordinary epidemiologic nose. I could tell you a number of extraordinary discoveries in his work, but this was the most important. He went after those birth records based on some other records that he had collected on the suspicion that there was a relationship between a child's birth weight and adults' liability for heart disease. The records were confidential. He couldn't get access to them. He realized his mother had grown up in Hertfordshire County, and he, in fact, was born there, so he convinced the county clerk, say, look, my mom's in that record. I'm not gonna reveal what she did nor anybody else. So he got the records. Turns out his mom refused even the 6'1" midwife and wouldn't have David or any of her other kids weighed. But he got the records, and in September 1989, published one of the most seminal papers in the field we're talking to you about. And what he showed is that the lower the baby's birth weight, the higher the liability for a range of cardiovascular illnesses and pulmonary illnesses. And of course, nobody believed it. It was scoffed at. There were many explanations. And even though it had been repeatedly replicated, here's a Finnish study which showed, this is the only graph you have to stand on your head to really understand. But this is birth weight here, low birth weight, but this is at birth, and this is higher birth weight by 11 years. And the biggest risk for cardiovascular disease comes from being low birth weight at the time of birth, but a rapid gain in birth afterwards. And the reason that, do you get the idea intuitively? Is that clear? That the highest risk for cardiovascular disease comes from a rapid catch-up when you're born low. Barker hypothesized that children undernourished prenatally adapt to or are programmed to live in an undernourished world. And the concept that even in fetal life, individuals adapt to an adverse circumstance and get programmed as if that adverse circumstance was going to continue throughout life is a pretty fundamental biological principle, which David Barker was rediscovering in his data. But despite these very persuasive data, it took animal studies, and you're going to hear about some magnificent ones in just a few minutes, took animal studies to clarify exactly the mechanism. And I'm just showing you one of what must be a literature of several thousand studies since then working this out. Here you have a study of rats where you can see that their birth weight in grams, and now we're controlling this experimentally by limiting the blood supply to the uterus in pregnant rats and the systolic blood pressure at the age equivalent of a rat adult. And you can see the higher the birth weight, the lower the blood pressure. So this was one crucial piece of evidence that has now been followed up with a variety of interventions and a variety of efforts to understand the mechanism. It established that there could be links between early adversity and the development of illness 5 or 6 decades later in individuals who didn't look abnormal, particularly when they caught up in their weight, from individuals who did not have that adversity. Here's a second line of evidence, and it's was presented first to the world by a remarkable professor of medicine at Johns Hopkins. You can see her dates. You can imagine what it took to break into medicine, let alone John Hopkins professorate. It took her until her mid-60s before she was promoted to full professor. She had, from her clinical experience, the sense that adult illnesses began early, and she wanted to spot the earliest manifestations of adult illness. Well, she didn't go back to the fetus, although there is perhaps something fetal about being in medical school, but she had a captive group of subjects, which were her medical students. And I'm showing your doctor maybe in this picture, if you look carefully, but she not only got the class of 51, but she got several preceding classes and several subsequent classes, and those medical students are being followed to this day. Many of them, of course, have passed away. Here's one of the most remarkable pieces of data from a follow-up well after Professor Thomas passed away. And this shows simply the difference between medical students who were born into families low in social class, mainly low income, and living in neighborhoods typical of low income families, and those who were born in favorable circumstances. Now, the reason this study had such an impact is that after you're in medical school, I mean, that's why a lot of mothers want their sons to go to medical school, is everybody's in the middle class, at least. So you're controlling adult social class just because they're all doctors, but they had a distinctive difference in their early childhood experience. And you can see already by the late 40s, those in the low social class are getting cardiovascular disease at a much greater rate and much more rapidly and much earlier than those in a higher social class. Now, we don't know if this is a prenatal or a postnatal adversity, but it draws, again, attention to early experience and late consequences. In building some kind of picture about how real this phenomena is, Steve and I have tended to give special weight to studies like Carolyn Bedell Thomas's study, in which children are observed as early as possible and followed through to adulthood. We've tended to give somewhat less weight, although I'll show you a couple of studies that use this technique, to retrospective accounts by adults as to whether or not they're abused, and I'll say a word or two about that. But here is probably the world's most important longitudinal study following children based in Dunedin in the South Island of New Zealand, whose investigators are Avshalom Caspi and Terry Moffat, following children from age 3 now until almost 40. And as you can see, this group is following early signs of ill health. Depression is, of course, itself an outcome, but also a risk factor for medical ill health. CRP is C-reactive protein. Your doctors may be getting that routinely in your lab scans. And then a series of factors called metabolic risk. And these are people who are not yet ill, but the effect of the social class in which they grew up, of definite maltreatment. Look at this. This is an unselected, random sample of New Zealand children and their families. Definite maltreatment, and this is probably an underestimate. So we're talking about a risk factor that's very serious and very prevalent. And then this is a more omnibus measure of social isolation of the kid and the family. But each of these risks has its own profile of emerging abnormalities that presage medical illness unless something is done about it soon. There's one other study we've rated heavily because it is also prospective, and this starts with a group of children with documented maltreatment and compares them with a very large sample of children who have no such documentation. And this is hemoglobin A1C, which is a sign of diabetes. You can again see that various forms of abuse and neglect significantly increase the risk for medical illness decades later. How could this happen? How could adverse circumstances of the kind I'm talking about enter the body and create a risk for medical illness? The simple answer is we really don't know, but there's some good possibilities. So I'm going to go through just three very quickly and then turn this over to Steve. Alyssa Eppel, who has been a central collaborator of Steve and me in our efforts to piece this puzzle together, as a very young clinical psychologist without extensive biologic training, but working in just the right place in the University of California, San Francisco Medical School, published an astonishing paper in 2004. This paper is approaching 3,000 citations, which is an astonishing achievement. She'd just been appointed an assistant professor. And again, I don't want to beat the gender equality bandwagon here too much, but Alyssa, after all of this, has just been appointed a full professor. What are telomeres? Many of you may know this, but you know we've got chromosomes that are shown here paired, and at the tip of each chromosome is something called a telomere, whose function is to protect the rest of the chromosome. When those telomeres begin to shorten, the chromosome and the cell in which it's housed is in trouble. It's liable for premature cell death. The body has a repair function, a particular enzyme called telomerase, and if you groove on scientific cartoons, this is what this is. It shows that when telomerase increases, it has the capacity to repair a shortened telomere. And Alyssa's first paper that I showed you in 2004 compared, and we're talking now about adult stress, compared mothers who were taking care of chronically ill children, ill for years, and if anybody in the room has had such an experience, you know how enduringly stressful that is. We're talking about the mothers and not the children, just for this first study, compared with mothers of healthy children. But what she found, and it took everybody's breath away, was the telomeres of, am I showing you the right thing? Yes, the telomeres of the women under high stress, and their telomerase levels were considerably less than those under low stress. In other words, it looked like, we couldn't be sure, but it looked like the chronic stress had worn down the tips of the chromosomes. So you can begin to get an idea how chronic stress in an adult could make a cell liable for premature death and the widespread impact of that on health. Now, it took a while to begin to connect that with child maltreatment, but in fact, and this study comes from Alyssa's lab, she found the exact same effects for adults who had reported maltreatment as children. So that's one possible mechanism. Now, here's another one, and I have to give you a quick tour of genetics, modern genetics. So you know that the chromosome is a lot of bundled DNA, and we're finding out surprises about DNA all the time, but basically the DNA has genes which, crudely speaking, we can divide into a promoter region which regulates whether or not the gene is expressed, an exon which contains, we used to think, most of the genetic, if not all the genetic information, and then what you've seen in the newspapers called junk DNA, the so-called intron, which we didn't think had much function, and there's a lively debate in the Times Magazine as recently as 2 weeks ago about this, but we're finding out many interesting things. But what happens in the ordinary course of biological development is that the DNA is transcribed into RNA. It's edited so that most of the intron, if not all of it, is taken out, and that edited or mature RNA leads to proteins, which are the fundamental building blocks of life. So here you're going to see another picture, I think, of Moshe Schiff. But here are two remarkable scientists who have produced an elegant collaboration, and they have actually shown, I mean, this was astonishing as Alyssa Eppel's work, that social factors can influence whether or not a gene is expressed. It was first done in a brilliant series of studies on rats, where they observed wide variation in the competence of maternal rats called dams in their care of their pups. Some dams were extremely effective, mainly in providing opportunities for the pups to nurse, whereas others made it very difficult for their pups to nurse. And what they showed was variation in maternal rearing of pups loosened a tiny molecule, a methyl group, and they've begun to trace out how, which lands not necessarily on the promoter, but very close to it, and silences genes in various locations. So you went directly from adverse maternal care in a rat model to the silencing of genes that were critical for normal, healthy functioning. Now, this has been now applied, this paradigm, to child maltreatment. I just want to say a word about retrospective reporting of child maltreatment. This is the most frequently used method. It's a questionnaire. It can be administered by an interviewer on a computer. It can be self-administered. It can be supplemented. But here's the kinds of questions that adults are asked about their childhood. Now, that's pretty straightforward. Can you read that? Well, try to touch me. Maybe pretty straightforward. That's pretty devastating, although infrequent. But there are also other questions. Is that child neglect? Is this the child's interpretation or an actual harsh treatment? Gives you an idea of one of the major questions driving us, which is, how do you know whether an adult has been maltreated? Now, these methods have been validated against clinicians' ratings, but we have found or learned from our colleagues in New Zealand that when you compare what adults say about their maltreatment and records, fastidious records, actually taken at the time of childhood, the correspondence is not great. So it's a major area of concern. But let's grant that this has some validity. And what you see here is, and you're going to see a lot more of this from Steve, is that the degree to which this methyl group, this little CH3, has blocked a critical gene that regulates nervous system function is directly related to the amount of abuse the adults in these studies have reported. And the reason I picked this study out, it's just published 2 weeks ago, is that this same methylation of the gene is strongly related to the functioning of an area of the brain called the hippocampus, which plays a central role in memory, response to stress, and health. So this is the sequence that this study suggests that from the experience of being maltreated to the silencing of a gene important for the functioning of a key brain structure, the hippocampus, to diminished size and function of this hippocampus is a possible pathway of how the outside adversity enters the body and sets up an affected individual for severe risk. So this is the transition slide to Steve's presentation. I think you've gotten some sense that as hard as we try and as clever as we may be in doing research with human subjects, there are a number of questions that are very difficult to tackle. Problems with accurate record, limits on the kinds of brain investigation we can do. For example, except in very unusual circumstances, if we're interested in the expression of genes, we can only sample various components of the blood, fatty tissue, and muscle. Fortunately, we don't sample brain tissue in humans and shouldn't, except in postmortem samples. But then you're talking about a huge number of steps between early abuse and a postmortem sample. There are limitations, as you'll hear, in brain investigation in primates as well, so rodents have a particularly crucial role in this investigation. But the point I want to make before Steve shows how his data begin to widen the circle of our understanding in this area is there's an absolutely fundamental problem in causal reasoning in human research. And just think of what we call one counterfactual. Think of a parent who's seriously and severely maltreating a child, neglecting the child, sexually abusing the child, beating the child. That is going to be a parent who is highly likely to have an addiction problem, to have their own mental illness, to have enormous problems controlling aggression. And many of those traits in humans, and I've talked about this before in a previous lecture in this series, are heritable. So in human studies, it's hard to distinguish when a child victimized by those horrible circumstances attain their liability because of the abuse or because they've inherited a number of traits that are not only not good for their behavioral health or their medical health. And that distinction is crucial for us to tease apart. And as you'll hear from Steve, animal research gives us a much clearer picture of what the maltreatment experience itself does to bodily function.
What about twin studies for the nurture versus nurture part of your question?
Twin studies could do it, but the prevalence of child abuse, although significant, you would have to have twin studies really effectively work here. You'd have to have one twin who was abused and one who wasn't. And so when you combine the infrequency of twins, that's one every 100th birth, give or take, depending on country and time and so forth. It's been hard to use the twin method to ferret out the nature versus nurture. It's theoretically quite possible, but we're dealing with one distressingly frequent event, but not frequent enough, and one relatively rare event, which is the frequency of twins. So you'd have to have a huge sample and be difficult, but I wish we could use that method. It's a good question. Yes.
Was there any possibility, or it seems unlikely, of a follow-up of the situation with the isolated, in separate cribs, Romanian orphans of a couple of decades ago? There was a case where children were brought up without physical contact, without being picked up or hugged. There was severe social.
Yeah, the question is, there are two major studies that have tried to learn as much as possible from the fact that in Romania, there were a large number of children who were brought up in orphanages, but a large number of them were brought to Britain and then raised in well-functioning families. Some remained in Romania, but a large number were brought to Britain. So what made that study or those studies very useful, was there was an on and an off. There was an on when the children were born and were exposed to multiple caretakers, none of them very good. Then there was an off period, which was when they were brought into a foster family. And one of the important outcomes of that study was a very distinctive way in which these children who had experienced multiple caretakers relate to any adult. I call it promiscuous attachment just to get people's attention, but it's indiscriminate. Those children will form a bond with any adult, which is very disconcerting to their primary caretaker and also to the adults who don't know these kids and find the kids latching on. The importance of that for this line of work is that, as Steve and I are looking at the data, we don't think we're going to be able to get a fully valid picture of which adults have been, and I've showed you some of those questions, so you got a feel for it, which adults have genuinely undergone maltreatment, which adults think they have, but maybe not have. Those adults, by the way, are at serious risk anyways, even if they haven't been maltreated. If you think you've been maltreated, that's also risk, but it's different. So we're thinking about other stigmata of early maltreatment, and are there behavioral patterns? And we've isolated two in the literature. One is this indiscriminate attachment, and another is a set of findings from a Minnesota longitudinal study that mothers who were sexually abused as children behave extremely seductively with their own male children. So we think that there may be behavioral indicators of early abuse that are especially manifest in the interaction between the adults with the abuse history and their own children. But that is barely a hypothesis. There's a little evidence for it, but those studies are extremely valuable, beginning to give us an additional repertoire of what we can think about in getting much more accurate estimates of which adult really had this experience and as a consequence is at risk.